Welcome to everyone, my name is Armando and in this video I want to update about my project within the ARCAID program. I am working on clonal isotyping of B-cell response in autoimmune diseases. The immune system is a complex network of biological process that protects our organism from diseases. There are many characters involved, among which neutrophils, macrophages, dendritic cells, T cells and B cells. The immune response can be divided in innate immune system and adaptive immune system. The cells of the adaptive immune system are the lymphocytes, the T cells and the B cells. There is a strong crosstalk between T helper cell and B cell in order to trigger an antibody response against a specific antigen. After this interaction, B cells start to differentiating in plasma cells that secrete a great amount of antibodies. Antibodies are composed of two kinds of polypeptides, the heavy chains and the light chain. Each of the four chains has a variable and a constant region. The variable region is encoded in several genes segments called variable, diversity and joining. The sequence variability is concentrated in three segments named complementary determining region. The most important for the antigen binding site is the CDR3. The CDR3 sequence can be considered as a B-cell clone fingerprint and it can be quantitatively measured, giving an idea of the expansion magnitude of a given clonotype. In our lab, we use the high-throughput sequencing technology to study the immune repertoires in autoimmune diseases. Sequencing the immune profile is clinically and diagnostically relevant. Specific B and T cell clones may represent a disease marker in a different individual with the same disease. Specific disease prognosis can lead to evaluation of similar CDR3s, may also be predictive of treatment response over time. With my project, we developed a new pipeline suitable for immune repertoire sequencing called RACE. .RNA is extracted from a wide range of tissues, including blood, lymph node, and synovium. The cDNA is retrotranscribed with a multiplex primer set. A template switch oligo that contains some molecular barcode is added to the reaction and incorporated in each cDNA molecules. Two consecutive PCR reactions designed with the semi-nested fashion are then performed using the cDNA as input. The final library is then sequenced with Illumina MiSeq that returns a lot of data that have to be analyzed. For the analysis, we use a bioinformatic pipeline developed by Clemson Group called Incantation Pipeline. This pipeline includes three important tools: Presto for the pre-processing step, Change.O for the database alignments and the reformatting table, and Alakazam for the data analysis. Thanks to this approach, we are able to sequence immunoglobulin repertoire, discriminating between isotypes and subtypes. We can detect the clonal frequency to identify high expanded clones, and moreover, this method is highly flexible. With few changes in the reverse primer sequence, is suitable to sequence different targets, including HLA, T-cell receptor, and other species. We examined and analyzed more than 10 samples in technical triplicates, and the results indicate that the pipeline is reproducible in both V genes percentage detection and clonal frequency detection. We are currently applying the method in several different studies. Of course, there is much more to say, but I will keep you updated about my project in the next video. Thank you for watching.